Okay, so now let's look into how a protein which is inside the nucleus can transport to the outside of the nucleus into the cytosol. So let's say we have this protein which needs to get transported outside the nucleus. And similarly, like the import receptor, we have an export receptor. So here we have an export receptor. So the export receptor would also have two binding grooves. Export receptor would also have two binding grooves where one, the cargo, the protein that needs to be exported binds, and the other, it binds to RAN in its GTP bound form. So RAN in its GTP bound form. So this is the cargo and this is the RAN. Now once these two are bound to the export receptor, let's shade it over here. Now when it is bound to both of these, then it can move out. So it moves out, and let's draw it once more. Now over here it is bound to RAN GTP, and it is also bound to the protein or the cargo that needs to be delivered. Now exactly when the import receptor was coming out, the exact thing happens. There is a molecule called the RAN gap, that is GTPase activating protein. So this is GTPase activating protein, and what it does, it activates the RAN, which is actually a GTPase protein. And once the RAN is activated, this GTP is hydrolyzed. This GTP is hydrolyzed to GDP and PI. So now it will be something like this. So here we have the receptor. And here now we have RAN bound to GDP and also the protein bound to it. Now once RAN is being hydrolyzed, the GTP on the RAN is being hydrolyzed to GDP, this would fall off. And along with that, the protein cargo would also fall off. So this would be the protein cargo, and this would be the RAN in its GDP bound form. So these two falls off. Now what happens, this export receptor or the exportin, it can move inside on its own through the nuclear pore complex. So this export receptor can move on its own through the nuclear pore complex, and again this RAN concentration, it starts increasing, or rather it starts decreasing inside the nucleus, and hence this RAN, this also moves in. This RAN moves in its GDP bound form when it is bound to another molecule called the NTF2. So NTF2 binds to RAN GDP and moves it inside the nucleus, and again this NTF2 can move out on its own. It does not require a receptor. So this NTF2 moves out and RAN is now in its GDP bound form. So again, the same thing happens like in case of the importin or the import receptor. What happened? This RAN GDP would be changed to RAN GTP by the action of a protein called GEF or RAN GEF. Now this GEF or guanine exchange factor would exchange this GTP and form RAN GTP. So RAN GTP. So this GEF or the guanine exchange factor would exchange the GDP that is bound to RAN to GTP. And again, this RAN GTP can perform another cycle of transport. So that is how the export of protein from the nucleus to the cytosol works.